What is life? For millennia, this question has haunted the most curious minds. In this episode of the Human Odyssey, we travel back in time to join the quest for an answer. Our story begins in the sun-drenched markets of ancient Greece, where the philosopher Aristotle first dared to look at the living world with a scientist's eye, classifying, dissecting, and trying to decipher the patterns of nature. His journey lays the foundation for a mystery that will endure for 2,000 years. We then set sail with a young, uncertain naturalist named Charles Darwin aboard the HMS Beagle. On a voyage to the far corners of the Earth, through volcanic islands and lush forests, Darwin uncovered a shocking secret: a mechanism of natural selection that sculpts all living things. But his revolutionary idea, born of lonely observation and deep thought, creates an even deeper puzzle. How is this information passed down through generations? The final act of our story is a high-stakes scientific detective thriller set in the 20th century. We enter the world of clashing ambitions and brilliant minds: Rosalind Franklin, James Watson, and Francis Crick, as they race to uncover the physical substance of heredity. The discovery of the DNA double helix is a moment that changes humanity's understanding of itself forever. A breathtaking climax to a story that began with a philosopher's simple, profound curiosity. This is the story of biology, a tale of how we came to read the book of life itself. That's what we're going to talk about next, so don't go anywhere. I'll be right back. The old man waded into the gentle surf, the Aegean tide cool against his ankle. To the merchants in the bustling agora of Mytilene, he was just another wealthy eccentric, a mainlander with a strange fixation on the slimy, crawling things the fishermen hauled from the sea. But Aristotle was not just looking, he was seeing. In his hands he held an octopus, its 8 arms a writhing knot of alien intelligence. While others saw a potential meal, he saw a marvel of engineering, a riddle wrapped in flesh. This, right here, was the beginning of our story. The year was 344 BCE on the island of Lesbos, and the question that drove Aristotle into the tide pools, the same question that would echo through the next 2.5 millennia, was deceptively simple. What is life? It's a question so profound it almost feels unanswerable. It's the pulse in your wrist, the unfurling of a fern, the silent predatory glide of a shark. It is the thing we are most intimately familiar with and the thing we understand the least. For most of human history, the answer was simple. Life was a gift from the gods, a spark of divine fire breathed into clay. It was magic, plain and simple. To dissect it, to question it, was to trespass on sacred ground. But Aristotle was a different kind of thinker. He was a student of Plato, but where Plato saw truth in abstract forms and perfect ideals, Aristotle was a man of the earth. He believed that to understand the world, you had to get your hands dirty. And so he did. He brought the octopus back to his workspace. With a bronze scalpel, he laid it open, tracing the path of its unique anatomy. He noted it had 3 hearts. He observed the strange branching veins. He saw how the male used a specialized arm, the hectocotylus, to transfer sperm to the female. A fact so astonishing, so contrary to all assumed knowledge, that it would be dismissed and disbelieved for the next 2,000 years. He did the same with hundreds of other creatures. He cracked open hens' eggs day by day, meticulously documenting the emergence of a living heart, the spiderweb of blood vessels, the slow miracle of a chick taking form. He was the first to see that whales and dolphins, though they lived in the sea, were not fish. They breathed air and gave birth to live young. He grouped animals based on shared characteristics, creating a hierarchy, a scala naturae, or ladder of life, with inanimate matter at the bottom and man, of course, at the very top. It was a first draft of understanding, flawed and biased, but it was a draft written from observation, not from myth. Aristotle was creating a new language, a new way of looking. He was the father of biology, not because he got everything right, but because he was the first to systematically believe that the secrets of life could be unraveled by looking closely at life itself. Yet, for all his genius, he stood at the edge of a vast, dark ocean of the unknown. He could see the chick develop in the egg, but he couldn't see what force was guiding its formation. He could classify 1,000 animals, but he couldn't explain the breathtaking diversity, the sheer, profligate variety of forms. Why 1,000 kinds of beetle? Why the peacock's tail and the viper's fang? His ladder of life was static, each creature fixed in its place by a divine architect. He had written the first chapter of the Book of Life, but the plot, the driving force of the story, remained a complete mystery. The clues were there, swimming in the sea and scuttling on the shore, but the detective who could piece them together had not yet been born. Two millennia passed. Empires rose and fell. Continents were mapped, and the stars themselves were charted in their courses. Yet the fundamental mystery of life remained locked away. The world was a library of unused books. The dominant story, elegantly simple and unassailable, was that of divine creation. Every creature, from the flea to the elephant, was handcrafted for its purpose, unchanged and unchangeable since the dawn of time. To challenge this was to challenge God, an act not just of scientific dissent, but of moral heresy. Into this world, in the winter of 1831, stepped a young man named Charles Darwin. He was not a revolutionary. He was a gentle, unassuming 22-year-old, a student of divinity who was far more interested in collecting beetles than in studying scripture. His wealthy father, despairing of his son's lack of direction, had called him a disgrace to the family. Charles was adrift, destined, it seemed, for a quiet life as a country parson. But fate, in the form of a letter, intervened. A former professor recommended him for the unpaid position of naturalist and gentleman's companion to Captain Robert Fitzroy, the stern, aristocratic commander of the HMS Beagle. The ship was embarking on a 2-year mission to chart the coast of South America. It was the opportunity of a lifetime, a chance for a grand adventure before settling down. With some trepidation, Darwin accepted. The 2-year trip would stretch to 5. And when Charles Darwin returned to England, he would be a different man, carrying a dangerous idea that would set the world on fire. The Beagle was a cramped 90-foot brig, a wooden nutshell tossed on an immense ocean. For Darwin, the first weeks were a blur of seasickness and misery. But as the ship reached the coast of Brazil, he stepped ashore into a world of intoxicating vitality. The rainforest was, he wrote, a chaos of delight. Everything was new, vibrant, and overwhelming. He saw iridescent butterflies, heard the guttural call of howler monkeys, and everywhere felt the hum of a world teeming with a ferocious competitive energy. This was not the orderly, peaceful English countryside. Here, life was a constant struggle. He watched a wasp paralyze a caterpillar, laying its eggs in the living flesh to be devoured from the inside. He saw armies of ants marching in merciless columns. Nature, he began to suspect, was not the benign, harmonious creation he had been taught to believe in. It was a battlefield. As the voyage continued, the clues began to accumulate. In Argentina, he unearthed the fossils of giant, extinct creatures. He found the bones of a glyptodon, an armored mammal the size of a small car. It was clearly extinct, yet its shell was uncannily similar to the shell of the modern-day armadillos he saw scurrying in the pampas. Why the similarity? It was as if one was a giant ancestral echo of the other. The idea of a static, unchanging world began to fray at the edges. But the defining moment, the plot point on which the whole story of modern biology would turn, came in the autumn of 1835. The Beagle arrived at a remote archipelago of volcanic islands straddling the equator, 600 miles off the coast of Ecuador. a desolate, sun-scorched place of black lava and thorny scrub, the Galapagos Islands. Here, Darwin encountered a world that seemed almost freshly minted. He walked among giant tortoises, so placid he could ride on their backs. He saw marine iguanas, imps of darkness, that sneezed salt from their nostrils and fed on seaweed in the frigid currents. But it was something much smaller, much more mundane that truly captured his attention. A group of drab, unassuming birds. Finches. He collected specimens from the different islands, barely noticing their significance at first. They were all finches, clearly, but they differed in subtle ways. Some had thick, powerful beaks, perfect for cracking tough nuts. Others had delicate, slender beaks, ideal for probing into cacti for insects. Some even had beaks adapted for using cactus spines as tools. It was only later, back in England, when an ornithologist confirmed that these were not merely varieties, but distinct species, that the full weight of the discovery hit him. It was as if one ancestral finch had arrived on these islands long ago and over countless generations had been sculpted into different forms to suit the unique conditions of each island. The nut-cracking finch didn't just happen to have a strong beak. It survived because it had a strong beak and passed that trait on. The insect eater survived because its beak was suited for its food source. The suspense was no longer in the adventure, but in the idea coalescing in Darwin's mind. It was a terrifying, exhilarating heresy. Life was not fixed. It changed. It adapted. Species were not created. They evolved. The mechanism, the engine driving this change, was what he would come to call natural selection. It was a beautifully simple and brutal process. Within any population, there is variation. Some individuals are slightly faster or stronger or better camouflaged. In the constant struggle for existence, those with a slight edge are more likely to survive and reproduce, passing on their advantageous traits to their offspring. The less fortunate, the slow and the weak, are weeded out. Over immense stretches of time, these tiny advantages accumulate, and new species emerge. Nature itself was the sculptor, slowly, patiently chiseling away at the raw material of life. Darwin returned to England in 1836, but he kept his theory a secret for over 20 years. He knew the storm it would unleash. He was a respectable Victorian gentleman now, married with children. To publish would be to invite ridicule and condemnation. He was, he wrote to a friend, confessing a murder. He toiled in private, accumulating a mountain of evidence from the breeding of pigeons to the structure of barnacles. The suspense of these silent years was palpable in his letters and notebooks. He was a man living with a time bomb. Then, in 1858, the fuse was lit. A letter arrived from a young naturalist named Alfred Russel Wallace, who, while suffering from a feverish delirium in the Malay archipelago, had independently arrived at the exact same theory of natural selection. The race was on, but it was a race Darwin had already secretly won. Urged on by his friends, he frantically condensed his life's work. In 1859, he published On the Origin of Species by Means of Natural Selection. The book was an earthquake. It shattered the old certainties. It replaced the comforting story of a divine plan with a new, grander, and more unsettling narrative of struggle, chance, and deep, deep time. It explained the finch's beaks, the glyptodon's echo in the armadillo, the wasp and the caterpillar. It provided the plot that Aristotle's story was missing. It gave us our place in the natural world, not at the top of a ladder, but as one twig on a vast branching tree of life connected by ancestry to every other living thing. But in solving one mystery, Darwin had created another, one that haunted him for the rest of his days. His entire theory depended on two things: variation and inheritance. He could see the variations all around him, but how were they created? And more importantly, how were they passed on? How did the parent finch transmit the secret of its beak to its offspring? He imagined some kind of gemmules, tiny particles shed by the body's organs that collected in the reproductive cells, but it was pure speculation. He had no microscope powerful enough, no chemistry advanced enough to find them. The great engine of evolution had been revealed, but its inner workings, the tiny gears and springs that made it turn, were still hidden in a black box. The secret of life, it turned out, was smaller than anyone could have possibly imagined. Now, the stage for the final act of our story is not a sun-drenched island or a teeming rainforest, but a series of cramped, austere laboratories in post-war Britain. The mystery is no longer what life is, or even why it changes, but how. How does a blue-eyed parent pass that blueness to a child? How does a bacterium pass its resistance to a drug to its descendants? The ghost in Darwin's machine had a name now, coined by a Danish botanist: the gene. It was the fundamental unit of heredity. But what was it? What physical substance, what molecule carried the immense encyclopedia of instructions needed to build a living thing? This was the holy grail of biology in the mid-20th century, and the race to find it was a tense, intellectual thriller fueled by ambition, rivalry, and flashes of transcendent genius. Our story converges on two locations. In London at King's College, there is Rosalind Franklin. A brilliant, meticulous, and fiercely independent chemist, she was a master of a difficult art called X-ray crystallography. The technique involved shooting X-rays at a crystallized molecule and recording the pattern as the rays scattered. It was like trying to determine the shape of a skyscraper by analyzing the way 1,000 tennis balls bounce off it in a hurricane. It was painstaking mathematical work. Franklin was tasked with using this technique to study the molecule that most scientists now suspected was the carrier of heredity: deoxyribonucleic acid, or, for short, DNA. Franklin was a perfectionist. She refined her techniques, producing clearer and clearer images of the DNA molecule. She discovered there were two forms of DNA: a drier A form and a wetter B form. And in May 1952, working late in her lab, she captured an image of the B form that was a stunning leap forward in clarity. It was designated Photograph 51. The image itself was a cryptic, ghostly cross of dark smudges. To a layperson, it was meaningless. To a trained eye, it was a revelation. The distinct X shape screamed helix. The spacing of the smudges on the cross gave the precise dimensions of the helical turns. It was the single most important clue to the structure of life, and it was sitting in Rosalind Franklin's desk drawer. 70 miles away, at the Cavendish Laboratory in Cambridge, were our two other protagonists: Francis Crick, a brilliant, booming 35-year-old British physicist who knew he was a genius, and James Watson, a gangly, ambitioned 23-year-old American prodigy who desperately wanted to be one. They weren't supposed to be working on DNA. Their lab had an agreement that the DNA problem belonged to the King's College group. But Watson and Crick couldn't resist the allure of the greatest scientific prize of their generation. They were an odd couple. Crick was the theorist, his mind a whirlwind of abstract concepts. Watson was the intuitive one, a visual thinker obsessed with the physical shapes of molecules. They didn't do experiments in the traditional sense. Their tools were conversation, speculation, and tinker-toy-like models of brass and wire built on their desks. They were trying to solve a three-dimensional jigsaw puzzle without knowing what the final picture looked like. The first attempt was a disaster. They built a model, a triple helix with a phosphate backbones tucked on the inside. They were so confident that they invited the King's College team, including Franklin, to come see it. Franklin took one look and, with withering precision, tore it apart. She pointed out that, based on her data, the water-attracting backbones had to be on the outside of the molecule, not the inside. Humiliated, Watson and Crick were ordered by their superiors to abandon their DNA work. The plot seemed to have stalled. Watson and Crick were sidelined. Franklin, meanwhile, was preparing to leave King's College for another institution and was summarizing her findings. The crucial data, including photograph 51, was still largely within her own domain. The climax of our story arrives in a series of furtive, almost cinematic moments. In early 1953, Watson visited King's College. Franklin's colleague, Maurice Wilkins, who had a strained relationship with her, showed Watson photograph 51. He did so without Franklin's knowledge or permission. For Watson, seeing the photo was a lightning bulb. The helical structure was undeniable. He went back to Cambridge, his mind racing. He now had the most crucial piece of evidence. Shortly after, through other channels, Crick obtained a report Franklin had written for a visiting committee which contained her precise calculations of the molecule's dimensions. They now had the stolen goods. The key pieces of the puzzle gleaned from Franklin's painstaking work. The suspense was electric. They knew they had a very short window before Franklin, or perhaps the great American chemist Linus Pauling, solved it themselves. They returned to their models. With the backbones correctly placed on the outside and the helical dimensions from Franklin's data, the structure started to take shape. But the core of the mystery remained. How did the four chemical bases adenine A, guanine G, cytosine C, and thymine T fit on the inside? How did it all hold together? The final eureka moment belonged to Watson. He was fiddling with cardboard cutouts of the four bases. He had been assuming that they paired like A with A, C with C, but it didn't work. The structure was lumpy and irregular. Then, in a flash of insight, he tried pairing them differently. He realized that A and T formed a pair of a specific size, held together by two hydrogen bonds, and C and G formed a pair of the exact same size, held together by three hydrogen bonds. It was perfect. A always paired with T, and C always paired with G. These base pairs formed the rungs of a twisted ladder, the double helix. Crick saw it instantly. It was, as he later said, beautiful. The beauty lay not just in its elegant, symmetrical shape, but in its profound implications. This structure didn't just explain what DNA was, it explained what it did. The sequence of the bases, the A's, T's, C's, and G's, formed a code, a language that carried the instructions for building an organism. And the structure itself suggested how it could be copied. The ladder could unzip down the middle, and each half could serve as a template for a new identical strand, allowing the genetic information to be passed on with perfect fidelity. It was the secret of inheritance, the mechanism Darwin had searched for in vain. It was the guiding force Aristotle had sensed but could not see. It was the secret of life, written in a four-letter chemical alphabet. In April 1953, Watson and Crick published their discovery in the journal Nature. Their paper was just one page long, a masterpiece of scientific understatement. Tucked away at the end was a single oblique sentence: "We have also been stimulated by a knowledge of the general nature of the unpublished experimental results and ideas of Dr. M.H.F. Wilkins and Dr. R.E. Franklin and their co-workers." It was a footnote to a revolution. Rosalind Franklin died of ovarian cancer in 1958 at the age of 37, likely caused by her extensive work with X-rays. She never knew the full extent to which her work had been used. In 1962, Watson, Crick, and Wilkins were awarded the Nobel Prize. Franklin, whose photograph 51 had been the key to unlocking the puzzle, was not and could not be included, as the prize is not awarded posthumously. Which is a shame, of course, but it is what it is. So, from Aristotle dipping his hands into the cool Aegean to Watson and Crick assembling a metal model in a dreary Cambridge office, the journey was complete. The question, what is life, now had a physical answer. It is a code, a vast, ancient, and endlessly edited text written in a language of molecules passed down through billions of years of trial and error. That code connects you to the finches of the Galapagos, to the fossilized glyptodon, to the octopus in Aristotle's hands. This discovery did not end the story of biology, of course. It opened 1,000 new chapters. We learned to read the code genomics, to edit it, CRISPR, and to understand how tiny variations in its letters can lead to health or disease. The Human Odyssey, the quest for self-knowledge, had turned inward into the very core of ourselves. We are, all of us, living stories written in the same language. We are the legacy of a 2,500-year-old question asked by a curious man on a Greek island. The answer to a riddle puzzled over by a reluctant revolutionary on a lovely voyage, and the keepers of a secret finally revealed in a ghostly image of scattered X-rays. The quest is not over. It never will be, but we now hold the book in our hands. And for the first time in human history, we are beginning to learn how to read it. And with that, my friends, we come to the end of our new episode from the Human Odyssey series, and this story was the story of biology. I hope you enjoyed it. I hope you learned new things from it. But of course, as I always say, this is just scratching the surface. If you really want to dig deep into these things and learn a lot more about these things, you will have to do it yourself. You will have to trust the painstaking, long process of knowledge, the necessary road to knowledge. Trust me, it doesn't live in those shorts that you watch. Those shorts are fine if you want to be entertained. But if you really want to learn something, you have to dig deep. But I know that I'm blowing my words in a hurricane, but I'll keep trying anyway. And with that being said, thank you very much for lending me your time, for listening to this episode. I hope you enjoyed it. Remember, if you want to have access to all the episodes that I create on English Plus Podcast, you can do that by becoming a premium subscriber on Apple Podcasts or Patreon. You can find the link in the description of the episode. I'm Danny, and this is English Plus Podcast. Thank you for listening. Remember, never stop learning with English Plus Podcast. I'll see you next time.